Hello everyone, I'm Ramanujam, and today I'm going to elaborate on an incredibly exciting research that I was part of in Launceston, Australia that inquired, does DTP vaccination affect influenza vaccine immunogenicity in the elderly? This project was conducted under the supervision of Dr. Katie Flanagan. First, I would like to start off with the background to our study. While we are aware that the purpose of vaccination is to provide immunity against disease targeted by the vaccine, recent studies have shown vaccines to influence immune response to other diseases in a non-specific manner. This has shown to be both beneficial and detrimental to health. For example, the Bacillus calmette-guerin BCG vaccine have shown in randomized controlled trials to reduce all-cause mortality and offer protection to non-targeted diseases. However, the Diphtheria-Tetanus-Pertussis DTP vaccine in infants has shown to increase susceptibility to infections and death in several epidemiological studies. Surprisingly, such heterologous or non-specific effects have been observed primarily in females, initiating strong responses with adverse events, but underlying biological mechanisms for sex differences remain unclear, and the research in this field in the vulnerable elderly has not been conducted. In fact, increasing age has been associated with impaired vaccine response due to their poor antibody and cell-mediated responses. The elderly have poor activation of CD4 T cells that are critical during influenza vaccine response. Also, they are more susceptible to infections due to defects in their innate and adaptive immune systems, as you can see in the bottom right picture. The elderly have chronic low-grade inflammation in the absence of infection, called inflammaging, which is associated with greater than normal levels of pro-inflammatory markers, such as the tumor necrosis factor TNF, interleukin-1, and interleukin-6. Inflammaging also mediates increase in immunosuppressive regulatory T cells to dampen pseudo-inflammation in the body, which are also seen in the immune response to DTP vaccine. Thus far, no published literature has investigated the heterologous effects of DTP vaccine. One theory is that DTP elicits a strong pro-inflammatory response with increased secretion of TNF due to the adjuvant alum. Under low TNF concentration, TNF receptor positive T effector cells are stimulated via T cell receptors under maximal cytokine producers, mediating pro-inflammatory responses. However, with prolonged interaction with TNF receptor 2, TNF impairs TCR signaling in both regulatory T cells and effector T cells, and selectively expands regulatory T cells to mediate immunosuppression. One way regulatory T cells mediate immunosuppression is by shedding their surface TNF receptor 2 as soluble TNF receptor 2. Since regulatory T cells have greater TNF receptor 2 than effector T cells, immunosuppression becomes dominant. The differences in TNF receptor 2 in T effector cells and T regulatory cells are thought to be responsible for the non-specific effects of DTP. As the heterologous effects of DTP in the elderly and its sex differential effects are unknown, given its impact on the global health, the current project focuses exactly on answering these gaps. The aims of the project is to investigate the heterologous effects of DTP under influenza vaccine challenge model and assess the inflammatory homeostatic response and the role of inflammation-reactive T cells, the TNF receptor-positive T regulatory cells, antibody and CD4 responses to influenza vaccine, and finally the vaccine-induced gene expression profiles. We hypothesize that DTP vaccination in the elderly will favor the expansion of immunosuppressive TNF receptor 2 positive regulatory T cells with immunosuppressive effects to other stimuli. DTP vaccine will negatively interfere with the induction of influenza-specific immunity. 3. The DTP vaccine will modulate pro-inflammatory gene expression to dampen inflammatory responses. And 4. Females will be more susceptible to these heterologous effects of DTP. Study design. The study design involves recruitment of 300 elderly adults over the age of 65 and 150 young adults aged between 30 to 50 years old, following specific inclusion and exclusion criteria, and will be randomized into one of the three vaccine groups: DTP followed by influenza vaccine group 1, 4 weeks later, and influenza vaccine alone group 2, and co-administration of DTP and influenza vaccine at the same time group 3. Vaccines will be delivered intramuscularly into the deltoid muscle. And you can see in this diagram that the red dots illustrate the follow-up for bleeding, and the blue triangles indicate the day vaccines are delivered. Aim 1. First, the blood samples of approximately 30 ml in total will be processed and frozen at minus 80 degrees Celsius vials for serum plasma and PDMCs, peripheral blood monocular cells stored in DMSO, and PBMCs stored in Pfizer for all visits for all study participants. To answer Aim 1, PBMCs stored in DMSO will undergo a phenotype analysis using 14-parameter fluorochrome panels and LSR2 flow cytometer for surface markers like CD3, CD4, and FOXP3. Analysis for cytokine production will be undergone by testing in alpha CD3 and alpha CD3 CD28 supernatant of stimulated PBMC cultures. Also, analysis for cytokine production and TNF receptor 2 positive to regulatory cell correlation study by stimulating PBMC with PMA ionomycin, and finally, analysis for suppressor activity by testing TNF receptor 2 positive and TNF receptor 2 negative T regulatory cells to suppress T effector cells and innate lymphoid cell proliferations and cytokines. The preliminary results from infants in Gambia administered with measles vaccine alone, measles vaccine plus DTP, or only DTP at 9 months is shown. In this diagram, we can see the TNF response to DTP only has decreased, and so have other cytokines, interferon gamma, interleukin 7, and the TNF to interleukin 10 ratio. This effect is enhanced in females with DTP only, as seen in panel E and panel F. Sorry, my pointer is now, I can't bring it to the diagram, so we can see in panel E and panel F that females show enhanced depression of cytokine production with DTP only, and all panels are shown 4 weeks post vaccination. The pilot study in mouse that has been injected with aluminum adjuvant found in DTP has shown to expand TNF receptor 2 positive regulatory T cells, but has no effect on regular T effector cells. Thus, these results illustrate that TNF responses are suppressed by DTP vaccine and TNF receptor 2 positive T regulatory cells are enhanced. To answer Aim 2, after processing the blood, we investigate the cellular responses by separating CD4 positive naive and memory cells and analyze them with 14-color flow cytometry panel for cytokines. After culturing them with irradiated antigen-presenting cells and live influenza vaccine. Also, we measure antibody responses using hemagglutination inhibition assay. The preliminary results shown here is from the same Gambia infants study shown before, but here responses to non-specific T cell stimulus with anti-CD3, anti-CD28, LPS, and CLO75 are tested for immune challenge. Here we see the depression of cytokine production under only DTP. Also, the effects are enhanced in females as seen in panel A and panel B. To answer Aim 3, after processing the blood, we obtain PBMCs stored in Pfizer vials for day 0 and 24 hours post-vaccination in all groups, and purify the RNA from PBMCs and reverse transcribe the RNA to obtain whole transcriptome, which which is analyzed by next-generation sequencing Illumina High Sequencing 1500 at Monash University, Melbourne. Discussion. Cancer preliminary and pilot studies indicate that TNAT responses are suppressed upon DTP vaccination. Such differences in vaccine response are observable, and TNAT receptor 2 positive regulatory T cells are enhanced with DTP. Indeed, these results are consistent with previous findings in the literature and warrant further investigation in the vulnerable population, the elderly. So the significance of our project comes from the fact that it is the world's first complete investigation of cellular and molecular mechanisms of non-specific and sex-differential effects of two commonly used vaccines in the elderly, the DTP and the seasonal influenza vaccine. The use of systems vaccinology with transcriptomics, metabolomics, and proteomics to answer our aim 3 will allow us to identify vaccine-specific immune response, critical for in-depth and understanding. This will allow us the potential to alter global vaccine policy, both in terms of timing and efficacy, and complete understanding of immunogenicity in later life, as in elderly. The completion of our study is expected by 2019. As you can see in this timeline, we're just starting to look at our aim to responses to influenza vaccine challenge and into patient recruitment. Thus, at this moment, we do not have results analyzed, but if the study satisfies our hypothesis, we are confident that we could influence vaccine timing by questioning when to administer DTP vaccine, example whether together with other vaccines before or after, and also the latent period to determine the exact timing to give DTP vaccine either before or after. We can investigate the adjuvant in DTP, either to reduce its efficacy or modulate its properties. Nevertheless, our results will have an impact on the global health, and especially in the elderly over the age of 60. Finally, I would like to thank Dr. Michael Radcliffe and Dr. Ilana Fish for selecting me for the Queen Elizabeth II Scholarship, Dr. Katie Flanagan for giving me the opportunity to work in Tasmania, the medical students in Australia, Liza, Isaac, and Avinash for enhancing my stay in Tasmania, and the BSI conference for allowing me to present my research. Thank you.